This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 419, recorded on December 9th, 2016. This episode is brought to you by CuriosityStream, a subscription streaming service that offers over 1,500 documentaries and nonfiction series from the world's best filmmakers. Get unlimited access starting at just $2.99 a month. And for our audience, the first 2 months are completely free if you sign up at curiositystream.com/microbe and use the promo code microbe. Hi, everybody. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me here today in New York City, where the sun is streaming in the window. There are just a few clouds in the sky, but it is really cold out, because up here in northern Manhattan, it's quite windy. Dickson, Despommier.
Hello, Vincent.
4 degrees Celsius.
Yep.
Feels like -1 because of the wind chill.
It feels pretty cold.
Pretty windy.
Yeah. Came on all of a sudden, too.
Yeah, it's been a week of freezing temperatures.
Yep.
That's it. We're into winter. Also joining us from Western Massachusetts, Alan Dove.
Good to be here. And it is cold and windy.
Yeah.
Yeah. Very windy. Yeah. Gusty. Northwest at 22, gusting to 30 is what they're reporting at Bradley Airport, and I believe it.
Not
was just
great flying weather, I take it.
No, not very pleasant flying weather. I was not up today and would not have been, or would not have been for long, perhaps.
Or you would have been somewhere else right now.
Yes. Yeah, actually, taking off in weather like this when it's really gusty and windy is not that big a problem. Landing is the problem.
Yeah.
So.
Yeah, even in those big jets, passenger jets, sometimes you have rough landing. They're going, whoa, 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 bam.
Yep.
No, Vincent, you don't realize that there is a pilot and a co-pilot, and the co-pilot often gets the charge of landing.
I understand that. I
do know
that.
He was a Navy man.
No, the co-pilot is getting practice.
A Navy man.
You can tell.
That hard landing is a Navy pilot.
You can tell when the co-pilot...
No, that hard landing is just the wind is tossing you around.
Well,
I was never in the Navy, and I make hard landings in freezing weather.
But I've heard it often said.
I just worry that the wings are going up and down. I worry at some point it's going to touch the ground. That would not be good, right?
That would be horrible.
Not be good.
Do they have some autopilot thingy that prevents that, or is that all manual?
There is actually such a thing as auto land, but it's extremely complex and only works at certain airports under certain conditions. And a lot of the training to use it is all about what to do when it fails. So the actual landing is almost always done manually. That's the one thing that is very, very hard to automate. And yeah, they do what they can, but sometimes the best you can do is to kind of slam it on the ground.
Also joining us from southeastern Michigan, Kathy Spindler.
Hi, everybody.
Hello, Kathy.
Hey, Kathy.
We have we have weather. It's 29 degrees, but it feels like 20.
And it's Fahrenheit, right?
Yeah, Fahrenheit. It's gray, cloudy, although I can see some blue sky and white clouds, so the sun is out in the distance. And so you were asking, I guess you wanted in Celsius. That would be -1 or so Celsius.
Also joining us from Central Texas, Rich Condit.
Hey,
is it
how you doing?
It's kind of north central.
Howdy.
Right?
Howdy, everybody. Yeah, we had this discussion before, and I decided some Texan may write in and disagree with me, but at this point, I have to say that I think it's probably closest to Central Texas.
Right. All right, let's go. I got it right here. It is a little south.
A little south and a little maybe touch east, but I think it's probably central more than anything else.
Well, we can placate the Texans by saying it's such a huge state.
That's right.
There's a big center to it.
I could just say Austin, Texas.
But you're in the frying pan,
do that.
Not in the panhandle.
You can do that. And Austin, Texas, people can identify with that, even if they don't know where it is. The name means something.
Yeah.
Is that where you have the bridge with the bats that come out? Is that Austin?
Yep, that's the Congress.
Have you gone to see it lately?
I've not gone to see it lately. It's apparently seasonal.
Mm-hmm.
Okay, these bats, I guess they migrate or something like that. I have not seen it lately. I have seen it in the past a couple of times. Right. By the way, it is 45 degrees and cloudy. That's 7 Celsius. So it's not real. That for me is cold. And it's overcast type cloudy. And I find it after 26 years in Florida annoying. Okay. But I don't know what normal is here now, and I don't think I want to know. So don't write in and tell me. And I'm not telling. I'm not even asking my daughter and her husband. I'll find out for myself. But actually, a cold.
I'm going to say you should have thought of that before you moved.
Now, there's only one thing worth thinking about, and that's being with my family. Okay,
there you go.
That's the whole deal. So at any rate.
Well, and his other options were what, Seattle and Boston?
Exactly.
Yeah.
Right.
Seattle's not bad.
I love the towns. Okay. The weather isn't great. And there's a small matter of money as well. Okay.
Right.
So, Alan, I wonder if you talked about it getting cold. We had a cold front go through here. Over the last 24 hours. I guess it's too early for that to reach you guys, and I don't really know whether what goes through here winds up going through you guys.
Not usually.
Anyway.
Not really. We get the stuff that comes across the upper Midwest and down.
Right,
and the Texas systems are more likely to go across the Southeast. So when you were in Florida, you were getting weather coming over across the Gulf from Texas.
Right.
But, of course, sometimes we'll get a storm that'll come up the coast. But mostly we get stuff that's come across Canada.
We get a lot of stuff out of the Gulf. The Gulf, and then it hits the Appalachians and comes up.
So at any rate, there we are. There's
that's it.
In meteorology.
TWiV.
I can hear the teeth gnashing.
That's right.
When are they going to talk about
get on with it, God.
Come on. The majority of the people that write in tell us their weather. Okay, I think most people are into it.
They do, but that's just because we do it. I think
it's become a thing.
Dickson, we didn't do it on the first episode.
No, we did not.
Came later. Not sure when, but
came much later.
All right, we have a follow-up from last week's episode. Drake writes, "Hello, professors. Dr. Dove qualifies in my book." Hey, you've been promoted.
Well, I was an adjunct for a little while.
That's right, at NYU, yeah. Yeah, you're better off not being a professor, actually.
I'm fine with not being a professor these days.
Right. It's very difficult. As I was looking over the MERS paper from last episode, I noticed that the sole contribution of authors Tang and Marasco was providing critical monoclonal antibody reagents as described in their 2014 PNAS paper. Producing enough antibody for animal studies can vary between a tremendous amount of work and a trivial amount of additional work, but I distinctly remember some journals state that providing reagents does not warrant authorship, and the NIH general guidelines state that authorship for reagents should not be given if they were already published.
Oh, my.
In this specific case, Nature Publishing Group's authorship policy does not provide specific guidelines. Authorship decisions can be very subjective, and since authorship acts as a currency in academic science, can have an incredibly influential impact on someone's career. Speaking generally, should professional societies or funding agencies attempt to encourage more objective standards or policies, such as authorship requires ownership, a greater than 50% contribution of at least one figure panel or job titles cannot disqualify someone from being an author. Some labs never give authorships to technicians. Okay, before we go on, I think we could have some opinions about this. I think the authorship should be up to the authors and no one else. I think if you publish something, though, and this is not the topic of this article, you need to share it. That's a separate issue. But I don't think the journals, I don't think NIH should say who should get what. I mean, if you give someone an antibody that they could buy, you shouldn't be an author for that. But the people writing the paper should know better. But if you've made something and it took years and it hasn't been published and it's precious, yeah. But again, it's up to the authors. Let them decide. What do we need people telling us what to do? Any other thoughts?
Yes. I gave
Thank you.
first authorship to a technician once.
Yeah, techs are fine.
And I got
Wow.
criticized on an NIH grant review by saying that I hadn't been the first author on a number of papers. That's actually
Why do you need
a good
to be,
thing, not a bad thing.
You don't want to be first.
No, listen, that was the criticism, but it was sort of reverse criticism, I thought.
That's ridiculous. I'm so sorry. Let's resubmit,
Yeah.
Dickson.
Well, the other thing is I've seen journals that actually require each author to list the percentage effort that they put onto
Yeah, now they do that more.
And that's a good thing, you know. In
Why?
this case, it would
Why?
be 0.1%.
That's ridiculous. Just put the authors on the bloody papers. Kathy, what were you going to say?
I was just going to say something similar to Dickson, but I think my first 3 or 4 papers as an assistant professor had my technician as the first author. She's the reason I got tenure. So, yeah, if people do the work in the paper, they should be an author. And you're right, it should be up to the authors to decide.
That's right.
Yeah, I did too. My techs were on papers early on as well. There's nobody else in the lab. Me and a tech.
Yeah, I had never really thought about this in concrete terms of whether or not somebody else should dictate what it ought to be. I've seen, and there's a link here. I don't know who put these links in. There's a link to the NIH guidelines on authorships at the end of this email. But certainly, we all just made the decisions ourselves. Okay, as you know, the lab decided and figured it out. And I put technicians on papers all the time.
You bet.
And as a matter of fact, there have been, part of my whole shtick was putting together a mutant collection and distributing the mutants. And I never, occasionally people would offer me authorship for giving them a mutant. And I would usually say that's not necessary. You don't have to do that.
Yeah. And
in only a very few cases did people actually go ahead and put me on the paper.
Mm-hmm. The thing that I objected to, Rich, was that when people wanted to put me on papers that I knew I didn't deserve to be on, but they wanted to use my cachet, so to speak, as a premier parasitologist, which I, of course, used to be, to enhance their own reputation. And I would refuse it and say, no, I don't deserve to be in your paper. All we did is have a discussion. Yes, but you made a very important, I just thought that was horrible to be in that position.
I didn't have that problem. I got no cachet.
No, I was tongue in cheek, please.
There was one incident years ago where I forcefully declined to be on a paper that I thought was a crappy paper. Okay, and I didn't want to be associated with it.
Right.
There are two. Well, so the thing about giving authorship to technicians, that is, there are going to be some laboratories where they just don't do that, and I think technicians should probably take that into consideration and maybe not work in those laboratories. But there's you have all come from and fostered a healthy environment around authorship, but there's a whole dark underbelly to this that involves really two kinds of offenses. One is what Dickson just described, where somebody busses in a big name to put it on a paper to kind of help maybe get into a more prestigious journal or make the thing catch more eyeballs. The other is people kind of foisting themselves onto a paper. We always put our department chair on as an author, even though he didn't do squat. And those are both very unhealthy practices that the NIH guidelines are trying to address. So this notion of honorary authorship or putting somebody on just because you want their name or just because they want their name on there, that's definitely a bad thing. I don't know that the journals are capable of policing it, and I do know that they're not especially enthusiastic about policing it. So, this monoclonal antibody, it was important to the research. Was it really hard to get or hard to develop? I don't know. So they made the call of putting these authors on the paper. I do think that should be primarily up to the authors. But I just wanted to point out that there is ugliness associated with this. And yes, that is a problem. And no, I don't have a good solution for it.
It's impossible to let the journals police anything because some of them say, if you publish in our journal, you have to share your reagents, and they can't enforce that at all.
Right.
People do not share, and they end up publishing again in the same journal because in the end, the journal wants the papers to get them more money.
Well, and there was just this week there was an incident in Science where they have published one of these the new thing now, an editorial expression of concern, whatever the heck that means, where someone had a paper published in Science that showed that microplastic particles were terribly harmful to fish. And it turns out that their original data were all on one laptop computer that got stolen from somebody's car.
Gee whiz.
At least that's the story. Right. There's also some discussion about whether those data existed in the first place. But the upshot, one of the things to come out of that is Science has a requirement, a stated requirement that when your paper publishes, the data behind it must be available to other researchers. And they must be uploaded to Science's website on their server in order to be available. Clearly, when this paper published, the data weren't available, but Science published it anyway. Mm-hmm. So if they have a policy about authorship or about anything else, to what extent is that going to be enforced when the paper looks sexy and is going to get headlines?
Yeah, right. Exactly. It's a business for them.
Yeah. Many of my technicians went on to graduate school afterwards, and they appreciated the fact that I included them into the loop, into the research, into the giving of papers.
Sure.
And the spirit of the lab was really quite high as a result.
Drake continues, changing subject. There is an interesting paper in Science that details a replication incompetent live influenza vaccine that is produced in cells with an expanded genetic code. Normal cells will be unable to produce full-length viral proteins due to premature termination at amber stop codons, while the vaccine production cell line will incorporate unnatural amino acids and translate the full proteins. So this is a very interesting paper. I think we should do another time, maybe next week. So I'll put that aside. And then he gives us a pick, which we'll do later.
So wait a minute. Is this an amber suppressing cell line?
Yeah. It's a suppressor. I don't know if it's amber, but it's a suppressor, right? You grow the virus and then you give it to people and they can't replicate in them.
Yeah, I really want to see that because is that linked to, because that was, I did a sabbatical messing with that many years ago.
We'll do it next week. Okay, a very brief Zika sitrep, a paper, Morbidity and Mortality, just came out today with a preliminary report of microcephaly potentially associated with Zika virus infection during pregnancy in Colombia, which is the country, not this university. Right. Dickson?
Colombia, not Colombia.
Right. That's exactly.
Approximately 105,000 suspected cases of infection during August through November of this year, including 20,000 in pregnant women. And so they have, this is the preliminary report of that, 476 cases of microcephaly compared with 110 during the same period in 2015. So this is a report that details what's going on there. Preliminary report. I'm sure we'll see something in the journals at some point. So there has been an increase, an apparent increase in microcephaly associated with Zika infection in Colombia.
And the timing of it is very interesting. It's 24 weeks after the peak of the epidemic.
In Brazil?
Which correlates with the emerging view that first trimester is the worst time to get this virus if you're pregnant. Mm-hmm. So 24 weeks, those kids would have been born.
Right.
If you got in the first trimester, 24 weeks later, the kid is born, and that's where your peak in microcephaly is. And the other interesting thing is when you look at the figure, by the way, of course, it's CDC government document, so it's open access. The peak in microcephaly is now tapering off and declining, which is precisely what you would expect to see with the decline in new Zika cases.
Right.
So as the population has gained herd immunity to this virus, and we're going to have probably much lower levels ongoing, the levels of microcephaly are declining at the same time. And that's all fits in with the view that has now come to the fore that this is indeed causative.
Yeah. We'll hear more about that, I'm sure.
Yeah.
Okay, now we have a paper for you, just published yesterday in Nature. It is entitled Host Genome Integration and Giant Virus-Induced Reactivation of the Virophage Mmivirus. And it's by Matthias Fischer and Thomas Hackel. And this work was done at the Max Planck Institute in Heidelberg. 2 authors. Matthias Fischer, we know for a while he's been working on big viruses and virophages. And I don't think he's been on TWiV. The reason I went to the Giant Virus meeting a couple of years ago in Germany and did a TWiV from there was because of Matthias. So Matthias sent this paper to you a couple of weeks ago, right, Rich Condit?
Yes, he did. Matthias and I have an association from that same ASV meeting where he was, using the ASV meeting to help organize his giant virus meeting.
Right.
That was the first time I met him, and we subsequently, out of that meeting, published a special issue on giant viruses in virology, where he served as a guest editor. So I've enjoyed my association with Matthias. He's a hard worker and gets it right. And actually, I was interested in this. He's very rigorous. That's one of the things I really respect, and answers your emails and that kind of stuff. I was interested in the fact that you don't often see anymore, in particular in a Nature paper, 2 authors.
Right.
Where the
other author is Thomas Hackel.
Yes. Where the person that you associate with as being, the lab head is the first author.
The first author.
This is relevant to our discussion of authorship. So I actually wrote Matthias and I asked him about that. And he said that this is basically his main postdoctoral work done during the last 4 years. He did most of the lab work himself with some technical help of the people mentioned in the acknowledgments. His mentor in the institute was not involved in the research, so Matthias did the planning, analysis, and writing. No complaints. And sometime during the process, he became a project leader, which I guess is more like a staff, more like a faculty position. And hired Thomas Hackel, co-author on the paper. He's a bioinformatician, and Matthias says not just a computational biologist. And worked on the Mmivirus project as a postdoc. He's an expert in next-gen sequencing data, and without him, I could never have assembled and interpreted the genome data. Okay. And he's now with Penny Chisholm at MIT for another postdoc. And he said subsequently, because I questioned him about not having his postdoc mentor on the paper.
That would be Curtis Suttle, right?
No, this must have been a second postdoc or whoever was his mentor in Heidelberg, because I think this was all done in Heidelberg.
I see.
Yes, he did a postdoc with Kurt Suttle, but I think he must have started in Heidelberg as not a lab head. And he said, it's the policy here that only people who contribute to the research get authorship. No automatic authorship for department heads. Plus, our director encourages independence of junior scientists. I would have gotten in trouble with my mentor if I had listed her as an author.
Wow.
Okay.
So she would have gotten her hackles raised.
Indeed.
Indeed.
He says, my position isn't exactly faculty equivalent yet, even though I now have my own research funds. I haven't been officially upgraded to group leader. Formality that may or may not happen, but doesn't change anything about the way he runs his group.
He's not yet Fisher 2.0.
Right. My virus projects have always been so exotic that I've done my own research since I came to Heidelberg.
Yeah.
And as long as I'm at it and talking about my correspondence with him, he wants the TWiVers to know that he has 2 postdoc positions available.
Yep. I got that.
And I imagine he'll get a flood of letters after we do this paper.
Because he clearly takes good care of postdocs.
Yeah.
So this is about virophages. Virophage, I think we've talked about them before on TWiV. They're parasites of parasites. Amazing. They are viruses that can only replicate in a cell infected with another virus, in this case, a giant virus. So I think the first one we talked about was Sputnik, which was found in cells, amoeba infected with Mmiviruses, and subsequently others have been detected. Now, they need a function provided by the helper virus, by the giant virus. And not what that is, I don't believe we know, but these viruses encode capsid proteins. They're not huge, but they have 30 or so kilobase pair double-stranded DNA genomes. And what's missing isn't clear, but they have to be in a cell infected with another giant virus. They actually inhibit the production of, so for Sputnik, it was shown a while ago that the replication of Sputnik inhibits that of the bigger virus, the Mmivirus, and it also allows the cells to survive. Now, the virus in this story, Mmivirus, we have also talked about because these Mmiviruses, they also require another virus infected cell to replicate in, but they're related. Their sequence is related to transposable elements called Maverick Polyton elements. So that's where the name came from, Maverick. So Mmivirus has sequence homology with Maverick. Now, these are DNA sequences, transposons that move around the genome. So they have enzymes that can cut them out and duplicate themselves and move them back in. And, who knows if they were the first, maybe they were precursors of viruses and they acquired a capsid and they left the cell.
So did they start as viruses or did they start as transposons?
That's right.
Do Maverick viruses feel the need for speed?
Yeah, right.
Yes.
They left home. So that's what these Mmiviruses are. And in fact, these DNA transposons, the Maverick Polyton elements, are found in lots of eukaryotic genomes. They encode a number of genes, including capsid proteins, these virophages, and integrase genes. And it's been reported that they can integrate into a variety of DNAs, including Mmivirus DNA. So these virophage DNAs have been found integrated in Mmivirus DNA genomes.
Wow.
It's so promiscuous. It's not the word I'm looking for, but it's
good enough. That's pretty good. I like that.
So this paper addresses this idea that these virophages can protect host cells. So what they do here is they co-infect. Now, Mmivirus infects cells that are infected with a big virus called Cafeteria roenbergensis virus. And C. roenbergensis is a marine phagotropic flagellate. So it's a marine cell organism with flagella, and it swims around the ocean eating lots of things, and that's why they call it cafeteria, because it eats a lot of stuff. And I remember,
and it's widespread and very active in the ecosystem, isn't it?
Quite.
So Cafeteria smorgasborgensis would have been a better double name, I thought.
And they are also on the buffet, so they're consumed in large quantities by plankton-eating organisms, which then feed into the whole food chain. So this is
amazing.
This is a major link. I mean, you don't see a lot of papers about C. roenbergensis because, for whatever reason, a lot of people don't study it, but it's out there, and it's a big part of the ocean ecosystems.
Yeah. So the virus, crow virus, that Matthias has been working on for a while, I think it was one of the biggest viruses out there, DNA viruses out there. So what he does in this paper, what they do, they co-infect C. roenbergensis with the virus and Mmivirus, crow virus and Mmivirus together. They take the cells that survive and they identify ones that have an integrated copy of the Mmivirus genome in them. Okay? And they do this by sequencing, and there's a lot of details here that we don't need to go into, but basically they find, I think, 11 different integrated. They pick one C. roenbergensis line, and it's got 11 different integrations of the Mmivirus in it. And
it's probably worth pointing out that one of the complexities here, and probably the major reason that there's a bioinformatician on the paper, is that when they did the genomic sequencing, it looks like roenbergensis is diploid.
Yeah.
And so the integrated spot for Mmivirus, there'd be one allele of that and the other would not have that, and it would mess up all the contigs in assembling the sequence. So it was very difficult to assemble out of that, and they had to do some tricks.
Right.
But they did find, in fact, these samples where you've clearly got an integration.
Right. So these are cells that are growing, they're healthy, and they have multiple integrated copies of the Mmivirus genome in them. They got the whole sequence, plus they found on either side of the integrated genome what they call terminal inverted repeats. There are 615 base pairs long on either side. They're very identical to each other. And I asked Matthias this morning where they came from. He said they don't know because it's not in the genome sequence, and maybe they missed it. They have no idea. It has no homology to anything.
Maybe Craig Vintner knows.
Or it's something that's maybe it's a product of the way Mmivirus integrase operates, because Mmivirus carries its own integrase, and that's presumably what's doing this.
Now they see that at each integration site there's a duplication at either end, and that's a feature of integrase. And when retroviruses integrate into the host DNA, you get a duplication of the target site on either side. But in that case, you get a loss of sequence from the viral DNA. You get 2 base pairs or so lost from either end of the viral LTR. This is for retroviruses. So this is very different in that sense. Okay, so they have identified integrated copies. You could call these lysogens, right? If by analogy with lambda infected bacteriophages, so they're not being expressed. And in fact, they look at that. They look to see whether RNA is produced in these cells, and it is not. But if you infect these lysogens with crovirus, then the genome starts to be transcribed.
You complete me.
Sorry?
It's another Tom Cruise quote.
Actually, your use of the word lysogen messes me up here. Okay.
Why?
I'm channeling Jane. Okay.
Sure, that's good. Good. Go ahead.
Lysogenic. The origin of that is I think lambda immediately.
Lambda.
Yeah.
And the origin of that is that you have a cell that carries a prophage, and that certainly applies in this particular case. But then that can be induced to replicate.
Right. Sure.
I suppose you could say this qualifies from the point of view that, as we're going to see, this is induced to replicate by infection. But I guess I just don't think of it in the context of an infection by another agent. I think of lysogeny as something where it can be induced to replicate on its own by some other environmental stimulus like UV light or starvation or something like that, which I don't believe is the case in this case.
And also that when it is induced, it kills the host cell. It goes into the lytic phase of its own life cycle, whereas Mmivirus is not going to kill C. robergensis, if I can say it. Because it's not capable of doing anything in the absence of the giant virus.
So maybe if one is a provirus, the other should be a semi-provirus.
It's an amateur virus.
It's an amateur virus. That's right. It's a pro-am virus.
So the way that these proviruses are induced is by infecting with crovirus. So there is complementing virus there.
Yeah.
So it can replicate. I believe it does kill the cells.
The crovirus kills the cell.
Crovirus kills the cell.
Right.
The crovirus kills the cell, yes.
So
we don't know what Mmivirus does by itself.
Nothing.
Yeah, that would be hard to know because you can't.
Doesn't do anything.
If you infect, it doesn't get to the end of the cycle, right?
Right. So how many proteins does the Mmivirus encode for?
It's about 20 kilobases, which would be enough for about 20 proteins.
Yeah.
Right. And the crow virus?
This huge.
Huge.
We're talking about megabase or something.
Huge.
Like that.
That's almost a cell.
One of the giant viruses.
Big one, much bigger. So you could object to lysogen, but
I think probably my objection is just based on my really, really strong association with lambda and other phage.
That's no problem. So what do you want to call them? You call this a provirus, but what is the CRO-V with an integrated Mmivirus? What do you want to call that? You don't want to call it a lysogen, huh?
Well,
not the CRO-V.
I don't have a better word.
Crow.
The crow. Yes. I'm adding a V there. Anyway, don't worry about it. I understand your objection. And I cannot object to your objection because I object all the time to other things.
Well, it's like if you have, a cell with an integrated retrovirus.
HIV or any retrovirus.
You call that a lysogen?
No, I don't. So you want to restrict lysogens to bacteriophages because that's where they originated?
I don't know. I mean,
the problem here is that these are called virophages, and they're not phages, right?
That's right.
Bacteriophages are viruses that infect bacteria. So
we need Judge Jane to rule on this.
This is a real problem.
Speaking of which, I just found out that you can't talk about the viruses that infect archaea as phages.
That's right. That's absolutely correct.
Because they're not bacteria.
Because they're not bacteriophages.
No, they're viruses. They're just viruses.
They could be archaeophages and just be phages for short, but no.
Yeah, because phage just means to eat, right?
Right.
True. So, yeah, it could be, but they don't want to be called bacteria. You certainly can't call them bacteriophages.
No, but you could call them archaeophages.
So maybe virophages is okay, right?
That has never been okay.
That's never been okay. You're right.
We've had that discussion before.
Yeah, virophage is no good because they're not eating another virus, right?
That's right.
Okay. Anyway, when you infect these, quote, lysogens, unquote, because I don't know what other word to use.
I think, for the purposes of the discussion, let's call them lysogens.
With crovirus, the genome is transcribed, it replicates, the DNA replicates, and new Mmivirus particles are produced. And this whole process is inhibited by treatment with cyclohexamide, inhibitor of protein synthesis. So it tells you that protein synthesis and also acetylcholine, is that a DNA synthesis inhibitor?
That's an RNA polymerase inhibitor.
RNA polymerase.
Okay, so that's inhibiting transcription from the Crow virus.
Yeah.
And so they presume from that some sort of early gene product from the Crow virus is responsible for activating the Mmivirus, and the likely suspect is a late gene transcription apparatus from the Crow virus. Okay, so the idea would be, now this is a stretch, and they admit that this is a stretch, but the idea would be that the Crow virus goes in and as is typical in these sorts of things, has some sort of early phase of transcriptional phase of infection, and then alters or synthesizes new transcriptional apparatus to do its late transcription, and it's that apparatus that's interacting with the Mmivirus genome, because there are some similarities, I guess, in promoter sequences and stuff. So it makes sense that transcriptional apparatus could activate through transcription the Mmivirus genome and get that rolling.
Mm-hmm. Does that, Rich, suggest that if you divided up the Crow virus into pieces and tried each piece inside of a cell by transfection with the Mmivirus already there, that you wouldn't get an answer?
Well, that's a really interesting experiment, Dickson, because that sort of thing has been done to do things like identify. The thing that leaps to mind is identifying the DNA replication genes in herpes virus, where Mark Chalberg introduced a replication-competent plasmid, an origin-containing plasmid, into cells, and then took the herpes virus genome and chopped it up into chunks and threw all the chunks in and showed that would activate the origin-containing plasmid, and then slowly but surely pared it down to 8 different pieces.
Interesting. Okay.
Gene-sized pieces. Okay, so, and in fact, God, I'd have to reconstruct this, but there was a similar bunch of experiments done with pox viruses, yes, to identify the late transcription factors.
Look at that.
Okay, so that's kind of what you're talking about. I don't know if you do transfections in C. Roenbergensis, okay?
Or you could add them on to the virus, right? You could just stick them on there and see if the elongated genome would work.
But at any rate, that what you're suggesting is not without precedent and not out of the question.
Okay.
Infection of the lysogen with Crow virus, so it induces transcription. It also induces DNA replication of the Mmivirus and the production of infectious particles. And so what they find, which is really the big finding in this paper, that this reactivated virus in other words, the Mmivirus produced from the integrated copy, suppresses the replication of the crow virus that was originally used to infect and activate that by 2 or 3 orders of magnitude, and the result is that the cell population survives. Not every cell, but some of the cells survive and go on to have an
and importantly,
life.
Not the cell that was infected initially that reactivated the Mmivirus.
Wow.
Right. This protection happens on a population scale, under conditions where you're looking at a low multiplicity infection.
Right. So, every cell infected by crow virus will die, but in the population, not every cell is infected, and the Mmivirus prevents release of virus from crow virus from infected cells. So you protect the population.
It basically inhibits the spread through the population. Yes.
I mean, if it didn't, of course, then this dinoflagellate wouldn't be a keystone species.
It would have been wiped out by the virus.
Exactly.
No, I don't know about that.
There would have been something.
There's always something
else. An imbalance would have been created.
Yeah, something else might have.
I mean, there's cycles. Like these Emiliania huxleyi.
Yes.
They grow, they bloom, and then their bloom's restricted by viruses, and they go through cycles.
Right.
And we don't know if similar effects are regulating those blooms. That would be interesting to look at.
It certainly would.
But there is, not everything gets killed by viruses in the ocean, for example.
Right, and also in the natural setting for this thing in the ocean, there's a big dilution problem. So a virus that kills 90% of this species would probably be unable to find any more hosts before it degraded, just broke down in the environment, just because they'd be spread by so many liters of water.
So, see, Dickson, the solution to viruses is dilution.
But in any case, this it's kind of an altruistic protection mechanism, is what they're getting at.
Yeah.
That the initial infection is going to be fatal to the initially infected cell, but this reactivation of Mmivirus is going to protect its neighbor cells and the general population, and provide something like a herd immunity.
There you go.
Yeah, so that's what they say.
Yeah.
They say it's like it is a kind of immune system. How did they describe it here?
Well, they draw analogies to CRISPR-Cas as well. Or do they draw the analogies, or was that in the
restriction enzymes or things like this?
An antiviral protective effect of provirophages and their hosts. So it's kind of it's interesting, Dickson. You think?
I think that's more than industry. Actually, it's complicated stuff that actually works.
Well, and it fits into this emerging debate about altruism. Classically, we've talked about altruism as an evolutionarily adaptive mechanism in complex organisms. You're protecting those who may help pass on your genes, and so you're more protective of those who are close relatives. But then you get down to a unicellular level of a free living organism, and you wouldn't expect, from the classical view, you wouldn't expect altruism to apply in that case, because if your genes and your neighbor's genes are not directly shared. But there's this example, and there are examples of things that resemble apoptosis happening in single-celled free-living organisms, and you have to wonder, well, there's got to be some evolutionary benefit to that, and so it suggests that altruism may be a much more fundamental evolutionary mechanism than we'd previously thought.
Yeah, I don't know. I mean,
I've always thought of the word altruistic as an anthropocentric
Yes, exactly.
word.
I was just going to say that.
It's hard to grasp it in nature.
I was just going to say that because maybe that's not the way to look at it, and that's why we have trouble imagining how it evolved.
Exactly.
Right.
Yes.
Exactly.
So I actually wrote a blog post yesterday, and I called it Altruistic Viruses. And this morning, Matthias wrote, and he said, you mentioned this, I think it would be good to make a clear distinction between virus and host with this term. For the Mmivirus, it would be better to refer to mutualistic viruses instead, where 2 or more partners live in a symbiosis that is beneficial to all partners. Mmivirus can persist as a provirus. Host cells get protection from CRO-V. Altruism is also at play here, but it's exhibited by the host cells. So he would say it's the host cells that are dying. Selfless behavior. So I agree. I think that is probably not the right word because it has too many human characteristic.
Yeah, well, one of your heroes is Richard Dawkins. And
not a hero. I just like the
remember the title of his book, for God's sake.
The Selfish Gene.
Yeah, exactly
yes.
right. That's offensive at some level.
Why
How can a gene be selfish?
How could a gene be selfish?
That's
right.
a human characteristic, right?
Of course.
So
but it is
Richard Dawkins is offensive at many levels.
it is preserving. It is
Thank you, Alan.
Well, it is preserving itself. That's the right.
Yeah, but
I agree that selfish is not the right word. But for a lay audience, it makes the point.
Oh,
right?
It sold 1 million copies, right?
No, I'm reading it right now actually, and I dislike very much the title.
Yeah,
and I dislike much of his writing as well because it's too anthropomorphic.
Yeah.
But he also says, by the way, that the whole green movement is useless and that we'll never be able to grow enough food to feed the world.
So what does he know?
Well, he wrote it in the 1980s. So
he says a lot of things.
He does.
Long before Dickson Despommier was
yeah, or anybody
the
else.
father of the vertical farm.
So, that's
right.
Also,
Matthias had another comment.
Now, when I wrote the original blog post, I wrote Cafeteria Roenbergensis virus, italicized, right, because genus, species, organisms you italicize. And he said, it's not nomenclature. It's not exactly everyone's favorite subject, and I struggle with it a bit myself. But as I learned from Jens Kuhn, species names within virus names are not to be italicized.
right.
So this has taught me not to put any host names into a virus name ever again.
Well, there's no genus and species. I mean, that's what he probably means to say. Right?
Well, there is. It's Cafeteria Roenbergensis virus. And I've heard this before, but I forgot, and I italicized it.
Is there another cafeteria species?
It doesn't matter. What's the difference?
Well, if it's the only one of its kind, then I think that it doesn't qualify as being part of a group.
Well, no. What's being
Cafeteria.
objected to here is italicizing. You should italicize Cafeteria Roenbergensis when you're talking about the organism.
Right.
But when you're talking about Cafeteria Roenbergensis virus, you should not italicize Cafeteria Roenbergensis, apparently, because that would be italicizing the virus name, and viruses don't get italicized names.
Yeah, well, that's true. As opposed to, of course.
So it's non-italicized Cafeteria Roenbergensis virus, which infects italicized Cafeteria Roenbergensis. Yeah.
That's perfectly clear.
As if life wasn't complicated.
Yeah.
That's exactly
so
right.
once again, I'm thinking pedant.
Yeah. Well, that's what nomenclature is all about, right?
Yeah.
So, Dickson, in light with altruism again, let me just read what Matthias wrote.
Please.
So this sacrificing to release Mmivirus to protect the population can be interpreted as altruistic, selfless behavior or kin selection. However, one has to tread carefully with this term when applied to microbes. It's attempting to anthropomorphize, i.e. to put some intent in the concept of altruism, which, of course, there is no need for.
So,
Right.
Just this morning, I got an email from Bridget, who wrote, looks like this week's edition of Nature yielded a paper worth discussing. The authors look at an integrated virus in the genome of a unicellular eukaryote. And in addition to the paper being cool, I think this would be a great opportunity to discuss giant viruses again, which I think we did a little bit. I'd like to also learn about virophages, which I think we did. I've never heard the term before. Some basics and background history would be interesting. Plus, talking about the paper could spark lots of ideas from the TWiV crew.
You did.
The authors compare the discovery in these protozoans to a CRISPR-like adaptive immune response, and they think about the ecology of the interactions. I'd love to hear your take. Thanks for the awesome podcast, and thanks again for all the company that keeps me going through long cell culture experiments. It is 8F-13 in Boulder, Colorado. I've been wearing tights under my jeans to label. PPS, be nice to Dickson.
Thank you, Vincent. Thank you, Bridget. Thank you, Bridget.
Wow, it's really cold in Boulder.
Yeah, well, that's very cold.
How long ago was that? Oh, no, that was just received this morning.
Just got it this morning.
Yeah.
It's colder in Boulder.
Okay.
All right.
You know, this episode is sponsored by CuriosityStream. They are the world's first ad-free nonfiction streaming service. They have over 1,500 titles, 600 hours of content, and it was founded by John Hendricks, who used to be with Discovery, so you know you're going to get real science shows. It's available in 196 countries. You can look at the movies on the web or on your TV that's connected to the internet with an Apple TV or a Roku or any other of those devices. What they have is a a wide variety of science and technology documentaries, also interviews and lectures, things like Stephen Hawking's favorite places. I should call it Dickson Despommier's favorite places. A brand new documentary in which Dickson pilots a CGI spaceship across the universe, making stops at vertical farms on different planets.
That's right.
Boy, that would be a good series.
I would love to do that series. Invite me. I'll do it.
Actually, it's Stephen Hawking piloting the spaceship, not Dickson.
No.
And he stops at his favorite destinations. Digits is a 3-part series hosted by Derek Mueller, who created the YouTube science channel Veritasium that explores online safety and security. Deep Time History, a 3-parter on the history of the universe, underwater wonders of the national parks. They have lots of cool stuff. You could just go to curiositystream.com and search for like science or virus or bacteria, and you can see what they got, what they have. They also have a very large or growing super high definition library, the 4K content, 50 hours so far. And all of this can be had not only for 2 months, but for free. But if you decide to sign up, there are monthly and annual plans available that start at
$2.99.
Thank you. Less than a cup of what?
Cafe.
Check out curiositystream.com/microbe. Use the promo code microbe during sign up to get unlimited access to the world's top documentaries and nonfiction series completely free for the first 60 days. That is 2 months free of one of the largest nonfiction 4K libraries around. Just go to curiositystream.com/microbe and use the offer code microbe at sign up. We thank CuriosityStream for their support of TWiV. We do have some email. And if you've noticed, listeners, dear listeners, if you have noticed, we have been doing kind of short papers for the last few episodes, and we're catching up on our email.
Good.
Yeah.
Good.
We're in the last 100.
No kidding.
Cool.
So let's do a few. First one is from Tari, who writes, "Greetings from a longtime TWiV and TWIP listener. One of the courses I teach is Introduction to Emerging Diseases, a microbiology class for non-majors. The topics focus on global, national, and local factors that cause and influence the emergence and reemergence of infectious disease now and throughout time. The course began fall 2001, just days before September 11th. There has been no shortage of current topics in the following year. It is quite fun to teach this course. During office hours early this morning, I got the mad tidies and started cleaning out piles of paper. Under a big pile, I found the attached paper. It was written by a former student in the course, now an RN. She remembered her dad worked on poliovirus when she was a young girl and decided to interview him about his work. I found her summary of the 2004 interview. I no longer remember the student's name. She was married, different last name, but I'm certain she would want to share it. By the way, my commute this fall is far more enjoyable as my new car is equipped to listen to podcasts.
Ah.
I started with episode number 1 of TWIP and will work my way through each. Listening to TWIP is making Minnesota road construction season and resulting delays a bit more bearable. Thank you and enjoy. Tari is in the Faculty of Biology, but I don't know where.
Nice.
All right, so I put a link to the document. This is the interview of her student with her father. It's called Summary of Procedures Involved in Modification of Poliovirus Type 1 for Oral Vaccine by Don Nelson. It's so cool. I looked through this a while ago. He talks about vaccines. 2 pages.
Yeah.
Cool stuff. I think this is a part of history, so that's pretty neat.
Nice.
So check it out. Okay, Dickson?
Yep.
Very cool.
How do I get out of this? Okay. All right. Tidies. Dickson, do you ever get the tidies?
Oh, are you kidding? Absolutely. Every time at the end of my fly fishing season, I get out all my fly boxes. I put the lamp on. I get a nice glass of wine next to me. It's about 7 o'clock in the evening. I put on iTunes. I close my door, and I start sorting flies for next year. It's fun. I just love doing that.
Wow.
Oh, my God.
I love
Make
doing
sure you
that.
don't drop them in the ointment.
Well,
I remember years ago, my wife was watching this Hercule Poirot thing.
Oh, yeah.
What is that?
Yeah.
That was a series.
yes.
And I walked by and someone walked in the room and said to Poirot, "What are you doing?" And he said, "I'm rearranging my stamp collection by size."
Exactly. In color or shapes or, it's fantastic. Really. It frees your mind up to do other things
Can you take the next email?
I will do my best. Anthony writes, House sparrows and influenza. The role of house sparrows is no surprise, since it was shown that domestic finches can spread H7N9, and then he gives a reference. I wonder if sparrows spread the disease to people. Children playing on swings, slides, etc., contaminated with bird droppings comes to mind. I wonder if new influenza viruses from Asia are a factor in the decline in the house sparrow population. And then he gives another reference. If this is true, then sparrows should be seen as the canary in the coal mine. Not the canary, but rather the sparrow in the coal mine in this case, I think. So FWIW.
For what it's worth.
Anthony writes, thank you, Alan, for that. I thought it was some misinterpretation of World War I. Okay. And then finally, someone else has written, sparrows are not native to the United States. Of course, they're not. They were imported from the UK, but they're everywhere. See the beak of the finch for more. And also...
And then I added, it's also mentioned in the sixth extinction.
That's right.
So a couple of our picks have...
Yeah, I just read the beak of the finch, and it's by Jonathan Weiner, who's a professor at Columbia. Mm-hmm. He talks about there's a chapter on sparrows, and they're not native to the U.S. It's a really cool chapter. It happened a while ago. I forgot the year, but it's a professor in a New England college. It was a huge snowstorm one day, and he was walking to class, and he saw on the snow hundreds of dead sparrows.
Really?
So he was an ecologist, an evolutionary biologist, and he collected them all, and he brought them back to the lab. I don't know when he got to teach his class, but he measured them all and weighed them all, and he made some conclusions about why they died, which I've now forgotten, but go read the book.
They were all inbred.
No. Some of them recovered, and I think I'm not sure if the bigger ones or the smaller ones died. Some cool thing. Anyway. It's neat stuff.
My biggest recollection of Sparrow Control was from China. They were defacing all the statues in Tiananmen Square and places like that, so they decided to trap them with mist nets, and they did. And the next year, they had one of the biggest outbreaks of Japanese bee encephalitis you can imagine, because the sparrows were eating the mosquitoes. So they had to take down the mist nets and let them all come back.
So in the U.S., they kind of outcompete and displace a lot of the songbirds. Sparrows and starlings are big problems in that way.
You're absolutely right.
But they were introduced as one of those daft Victorian activities. There were acclimatization societies around the U.S. whose purpose was to bring novel species into the country and acclimatize them to our environment.
Indeed.
Was, as we know, not a terribly wonderful idea. It worked spectacularly well in some cases, and that's the problem.
A paper just was published in Scientific Reports in November entitled Surveillance for Highly Pathogenic H5 Avian Influenza Virus in Synanthropic Wildlife Associated with Poultry Farms During an Acute Outbreak.
Why?
Dickson, do you know that word?
I remember hearing it, but I can't recall its exact meaning as we were speaking.
Synanthropic means a member of a species that lives near humans.
Lives in close association with humans.
Humans, right. Okay. Anyway.
Not to be confused with lycanthropic.
Well, I would say peridomestic is another way to say that, too. Though.
Is that right?
Yes.
I like peridomestic.
Yeah, I do, too.
Anyway, they collected 2,627 samples from 648 individual birds and mammals. They didn't find any virus in sparrows.
Right.
But they were commonly captured. But these are for avian influenza viruses. Who knows about others? Mm-hmm. And it might have been done. What's the sixth extinction?
Alan, go for it.
Oh, that was a book I picked a while ago. That was it's about the mass extinction that's going on right now.
Oh.
And it gives a quick review of the previous 5 that were caused by
That's right. I remember that now.
Comet strike and volcanic eruption and that kind of thing, and then the current one and how it's unfolding. And one of the initial discussions is how we've kind of recreated Pangea by bringing together all these species that have been separated by millions of years of evolution, and we've mixed them all over the planet with often disastrous results. And that is one of the major causes of species getting wiped out, is that they're getting driven out or eaten or outcompeted by the introduced species.
Yeah, I have to tell you, I have a friend who's a geologist that I do a lot of fishing with. He worked up at Lamont Labs at Columbia, and at one point he had a bumper sticker that said "Help reunite Pangea,"
And we have.
Kathy, would you take the next one, please?
Sure. Scott writes, "Hi, Vincent and TWiV team. I'm writing to you from beautiful Tasmania, coincidentally in the very building where the devil facial tumor was first identified. Firstly, I just wanted to thank you for the excellent podcast. My career in biology has been quite varied up to this point, and it's only fairly recently that I found myself in a virology role. This has been very challenging at times because my background is in molecular biology and bacterial ecology, and until I started my current job 2 years ago, I had no experience at all in virology, eukaryotic cell culture, or immunology. I'm now working as a research virologist in a lab that develops diagnostics and vaccines for the aquaculture industry here in Tasmania. We're working with a handful of novel and interesting viral pathogens of fish. I won't give too much away at this stage, but we plan to have some work published in the next few months, so I'll be sure to update you with the details. When I took up this job, I moved to Tasmania, a small island state at the southern tip of Australia, from Brisbane, in Australia's northeasternmost state. During the 2,000-kilometer drive down here, I had plenty of time to listen to the Virology 101 series and try to bring myself somewhat up to speed on basic virology. This was incredibly helpful at the time, and since then, I found the TWiV podcast to be an entertaining way to keep in touch with current advances in the wider field of virology while setting up and reading countless virus culture plates and TCID assays. I was particularly interested to hear about the discovery of a novel orthomyxovirus of tilapia in TWiV 384. I try to keep on top of recent findings in fish virology, but that particular paper hadn't appeared on my radar until I heard you discussing it on the podcast. Given the current worldwide boom in intensive aquaculture, I suspect that there will be more and more stories of novel viruses showing up in fish in the coming years. Many aquaculture systems, including the ones used here in Tasmania to produce salmon, involve growing the fish in large cages that are anchored just off the coast. This means that the farmed fish are in close contact with wild fish and also birds and other animals that are attracted to the food pellets, so there are plenty of opportunities for wild viruses to jump from the environment into the farmed livestock. At least in this field, we are in the fortunate position that there are many established fish cell lines, so it's possible to culture a wide range of viruses. I wanted to ask if you are aware of any similar podcasts or online, preferably audio format, courses that deal with basic vertebrate immunology. This is still a major gap in my training, and it would be great if there was an immunology equivalent of the Virology 101 series that I could listen to while working at the lab bench. I've searched, but so far I haven't been able to find anything. Thanks again for the great podcast. Regards, Scott. P.S. It's been a long, wet winter here with lots of flooding, but we have beautiful spring weather today, 8C and sunny this morning, and it feels like summer isn't too far away. P.P.S. In one of the episodes where you were discussing the devil facial tumor disease, Dickson had suggested that devils were the only native carnivore in Tasmania. I noted that in a couple of later episodes, several listeners had correctly pointed out that we also have quolls here. I hate to anthropomorphize, but I think the quolls would have been a bit insulted by your comparison of them to rats. There are actually two species here. The larger spotted tail quoll is about the size and shape of a large domestic cat. It's a generally very shy nocturnal predator of birds, small mammals, and reptiles, but it is known to occasionally break into people's chook sheds, American translation, chicken coops, right. And eat chickens. The smaller eastern quoll is now extinct on the Australian mainland, but relatively still common in Tasmania. My understanding is that it is a bit more omnivorous and feeds mostly on invertebrates. It's about the size of a chihuahua with a bushy tail and is undoubtedly one of Australia's cutest native animals. Both species have beautiful spotted fur and really don't resemble rats.
No, so I take it all back, of course, again. Yeah. This goes underneath a whole list of misstatements. I just misspoke, okay? Misspoke.
It's a Dixonism, right?
A Dixonism, that's right.
For the immunology podcast, how about autoimmunity?
Yeah, that's what I was going to say.
Yeah, except that if you want like a course, that's not going to do it. Right. They tend to just do papers, and they tend not to be very frequent.
Yeah.
There is a need for a fundamental one for sure.
They should do Immunology 101 on autoimmunity.
Yeah, there you go.
I don't think they're going to do that, frankly.
Okay.
Rich Condit, can you take the next one, please?
Richard writes, you have probably already read the information contained in this article. Apparently there is a book available that chronicles all the testing. Over and over again, the military has conducted dangerous biowarfare experiments on Americans. This is from Business Insider.
That's a great resource, by the way.
Dateline San Francisco, Justin Sullivan Getty images on September 20th, 1950. A U.S. Navy ship just off the coast of San Francisco used a giant hose to spray a cloud of microbes into the air and into the city's famous fog. The military was testing how a biological weapon attack would affect the 800,000 residents of a city. The people of San Francisco had no idea. The Navy continued the tests for 7 days, potentially causing at least one death. It was one of the first large-scale biological weapon trials, and he links to the full story, which is in, as was said before, Business Insider, in a subsection called Science. Well, that's pretty gross.
Well,
and apparently doctors noticed outbreaks, an outbreak of what is this? Serratia marcescens, which had no reason to be there. And they were,
Yeah, and it's easy to identify because it's pink.
Yeah.
But they did that in the subway one year also, a long time
ago. This is true, right, Alan? They did this.
Yes. I actually learned about this in my college microbiology course. Our professor talked about the releases and S. marcescens, and there was one other generally believed to be harmless bacterium that was used in a lot of these. Bacillus subtilis.
Yeah, I was just going to say that.
And they're generally harmless, and what was being looked at was how would a bacterium spread through the air if released in the ways that biological weapons would be. But yes, of course, this was all classified and nobody was told about it. And we now would hopefully not do such things. But this did, in fact, exist.
Yep. Hopefully we're not doing this anymore, right?
Hopefully.
Hopefully.
Who knows?
Yeah.
Who knows what the government is doing and will do in the next 4 years? The next one is from John, who writes, "Hello, sir. My name is Joel, and I'm 15 years. I got very interested in the virology classes taken by you and came across synthetic virology. But I wasn't able to find information on it and how to become a synthetic virologist. I would like to know more about a list of universities that offer courses on synthetic virology."
Well, you start at your toes, putting yourself together as a virologist.
Yes, how to make a virologist. So, Joel, why did I write John writes? I'm so sorry, Joel.
Mm-hmm.
You came to the right place because I did synthetic virology a long time ago, and everyone uses it nowadays, basically. You could go to any university with a virology program, and you'd find some synthetic virology, which, of course, simply means that you make a genome of a virus and recover the virus from it. Now, technically, synthetic, what they mean is that you're going to synthesize the sequence of the genome and recover the virus. I would say that if you cloned it and altered it in some way, you could also be a synthetic virologist as well.
Synthetic biology is this term that gets thrown around. A lot of what is done under that term is what we folks in my generation and prior generations would have simply called molecular biology.
Yeah.
But apparently that's not cool enough anymore, so it's got to be called synthetic biology, but it's really the same thing.
I think I mentioned synthetic virology in one of my lectures, so probably that's where Joel, because I wanted to say this is not new, this synthetic biology. Virologists have been doing it for years, and they were among the first, right, to make viruses. So you can find, and Kathy has put a link to a review article. I believe that's, synthetic virology, engineering viruses for gene delivery. So that's one way you'd want to apply this to modify viruses so that you can do gene therapy and so forth.
Basically, anywhere you go to study virology, you're going to wind up jerking genes around.
Jerking genes around.
Right.
I like
and,
that.
That'll make you happy.
Is that like beef jerky?
Used to be gene jockeys. You could ride them or something, but this is different.
Dickson, can you take the next one?
I can. Jonas.
Take a deep breath. Take a deep breath.
Jonas writes, hello, my name is Jonas, and I have written a research proposal on a hepatitis C virus that I studied from the net, but can't find free publication or a journal that will accept it because they say it's not in their scope. And I'm disabled, so I have no money to do anything with it. Plus, there's other variables as well, and I don't have an institution backing me up. Is there anything I can do or showcase it on your site or show? I would appreciate any help or advice. Thanks, and hope to hear from you soon. Jonas.
I don't know what to do about this.
Yeah, what do you do about that?
I mean, I know you need money to put, even in these open access journals like PLOS ONE, you have to pay thousands of dollars to publish it.
Especially in the open access.
Exactly.
Yes.
And it's not going to get into a non-open access journal.
A research proposal.
My suggestion, Jonas, is to put it on your own website.
It's a proposal. Yeah. It's not a paper of research.
Yeah, it's a research
proposal to do research.
Right.
That's
so, Jonas, here's the thing. I don't know what you want to do with this.
Right.
But if you want to do the research, it's going to be really hard if you haven't got the proper training and an environment to do it in.
Yeah, exactly.
I don't want to discourage you, but I would say, put it online and maybe you'll get some critiques and people can help you make it better. It's not, you could do a free blog, right, Alan? You don't have to pay anything.
Absolutely. Go to wordpress.com and set up a free blog and put your idea up there. And if you want to send us a link, we can look at it. And I don't know if we'll be able to do anything about it, but you can at least put it online. Anybody can publish anything online, as we should all now know.
Unfortunately.
Yes, unfortunately and fortunately.
Yes, both.
Okay, Kathy, you're next.
Okay, Peter writes. Starts off with the URL, so I can't quite read that. Would love for you to talk this over at some time. I work at a government research funding agency in Sweden, looking into metrics to evaluate scientific excellence at the institutional level. Will H-index hold over time, or is there a need for adjustments given increasing prevalence of preprint servers with access logs and other measures than printed citations, etc.? Thanks for a great show. The weather in Stockholm at Karolinska Institute, home of the Nobel Prize, is completely overcast and 10 Celsius with a drizzle of rain forecast for later this afternoon. Cheers, Peter. So
could you define will H index?
It's also dark.
Yeah.
Sorry.
Can you define that for the listening public? H index.
Oh, H index is an author-level metric that attempts to measure both the productivity and citation impact of publications of a scientist or scholar. It's based on the set of the scientist's most cited papers and the number of citations they have received in other publications.
Home of the Nobel Prize. Well, that's how they do that.
That's the classic, impact factor, right?
Yeah.
Right. So this is it's one of many efforts that people have made over the years to try to reduce scientific publication productivity to a number that can then be scored, and everybody can be ranked by it, and we won't have to have any of these pesky debates about who's the bigger, better scientist. I think the H index is more recent than some of the older ones. Of course, yes, that would be the definition of older ones. The H index is just from a few years ago, 2005, I think is what Wikipedia says. And Jorge Hirsch came up with it. And so Hirsch is the H in H index.
I agree that we do need to have something better, because with everything online, you get downloads now. And over at PLOS, they're displaying article views and shares, and I think that's going to have to be factored in.
Yeah.
In fact, I'm linking to an article in PLOS ONE where they have something called the PR index, using the H index and PageRank for determining true impact. So it's a combination of both. So probably, citations plus page views is going to be important in the future. So I do think you are right, Peter. Mm-hmm. Rich Condit, can you take Charles's, please?
Charles writes, Dear TWiVers, in York, England, today. The weather is a mixture of heavy rain and sunny periods with the temperature currently 14 C. On the subject of Celsius versus Fahrenheit, I would like to offer this sketch from the BBC radio comedy John Finmore's Souvenir program as a listener pick. We're getting ahead of ourselves, but that's okay. Please excuse the dig at the end. I'm sure it's meant affectionately. And I actually listened to this
it's good.
YouTube video, and it's pretty good. It has Messrs. Fahrenheit and Celsius meeting at a party and comparing their
is it hot in here, or is it just me?
Yeah. Although I've never formally studied biology, I managed to pick up quite a lot in the past few years, mostly from listening to your podcast, followed up by a bit of research with Google to learn more detail about the subjects that come up. Of course, having to work out their spelling from an audio recording isn't always straightforward, and there is sometimes a discrepancy between what you say and what I think you've said. Some, like cow pea mosaic disease, that's cow PEE, or chicken gunya virus,
right,
were sufficiently dubious to raise my suspicions, whereas toe-like receptors conjured up such a plausible mental image that it wasn't until you explained the etymology that I realized my mistake. Memory glands also sounded possible, but I felt sure like that with a name like that I would have heard of them already. Turned out I had. I tell you, this guy's so
I wish
funny.
I had a few more.
This guy's so funny that when he finished this, I had to look and see if he was the comedian
Yes.
that did the Celsius versus Fahrenheit thing.
That's great.
You okay, Dick?
Memory glands is going to live on.
Frankly, I'd like to thank all the Twix team for the work you do in promoting science. In an era of increasing dogmatism, it is refreshing to be able to listen to reasonable people engaging in rational discussion. Kind regards.
Riot.
There you go.
Just to set the record straight for somebody who might be interested in cow pea mosaic virus, it's COWPEA.
Right. Yes. And it is not a disease of cattle. It's a disease of a plant.
No, and I actually gave a bad quote there from Celsius talking to Fahrenheit, who actually said, is it hot in here or is it just you?
One more round of email. Katreya writes, hi, Vincent and the rest of the TWiVrific gang.
Wait, did Kathy go?
Did you go? Yeah, you went.
Oh, you did? Okay. I lost track of the rotation, sorry.
First off, I wanted to thank you for allowing my fiancée and I to sit in on a recording of TWiV. We both had lots of fun. I also wanted to thank y'all for such a wonderful podcast. I started listening to TWiV about 5 years ago during my last job helping to manage the stem cell core at the Gladstone Institutes. Over the years, I've learned a lot from all the Twix podcasts, and they've inspired me to continue my studies after a 15-year hiatus. After moving back to Cleveland and landing a great job here in the case genetics department, I'm now dipping my toe, would that be a toe-like receptor? Into grad courses here with the intention to apply to those master's programs in systems biology and bioinformatics sometime next year. So thank you. Now, on to the question I had. You mentioned in the last TWiV how DNA tumor viruses utilize various oncoproteins to ramp up the host cell's DNA replication machinery for their own ends. This can have the effect of increasing cellular proliferation. Are there DNA viruses or other virus types that don't utilize this type of system, but instead modulate the host cell's enhancer landscape to positively influence either their own replication potential or cellular proliferation? I'm thinking of this in the context of viral protein products either directly or indirectly altering chromatin accessibility and regulating histone methylation, acetylation marks at regulatory elements associated with cellular proliferation. What are your thoughts? That's a great idea. Does anyone know of, I love this, the enhancer landscape. Is this a way of getting cells to proliferate? I'm not aware of it.
The first thing that comes to mind, though it's not directly relevant, is the studies with herpes virus, the control of latency, where the herpes virus genome in a latent state has a different set of chromatin modifications and presumably activation of the latent genome to go through lysis or a lytic cycle involves modification of those chromatin marks, but that's on the viral chromosome. What is it? Ketria is talking about modification of the cellular genome. Yeah. I also have a vague recollection of reading stuff about modifications that are associated with the DNA damage response and et cetera during virus infection. So, no, I can't think of anything. Those are things I think of where there's chromatin modifications that are involved in virus infection, but nothing specifically like exactly what she's talking about. Cellular modifications that the virus is initiating. But
now, I saw a recent paper where they did transcriptomic analysis of, I think it was cytomegalovirus infected cells, and there's a huge modification of the transcriptome of the cell in those virus-infected cells. Now, the mechanisms could include enhancer landscape modulation or other promoters or other things as well. So I think we'll find out. I think it's a good idea that maybe, kicking DNA replication isn't the only way to do that.
I've just pasted in something that's tangentially related. It's a Nature paper from Matt Weitzman's lab that came out this last summer, basically showing in adenovirus infections that a core protein binds to host nucleosomes and that sequesters immune danger signals. So the final line of the abstract is, the study uncovers a viral strategy in which nucleosome binding is exploited to control extracellular immune signaling. So it's not exactly what she's asking about, but it is a connection between viruses and chromatin. Yeah,
the potential's there. Right. She has a science pick. Am I reading this? Yeah. There's a cool YouTube channel called Periodic Videos featuring a group of chemists from the University of Nottingham talking about a myriad of chemistry-related topics for a lay audience. Anyway, keep up the good work. I hope to visit again sometime. Katreya is a research assistant at Case Western Reserve University, which is in Cleveland.
It's a good school.
Dickson.
Marion writes, "Hi, Professor. I'm Marion, a design student at the Art School of Geneva. Head Geneva. Geneva. And then lists a...
Don't put an R in it. What the hell are you putting an R in it? It's a Genève.
It's a French thing.
Genève.
It's pronounced Norwalk.
Exactly. That's Geneva, New York, not Geneva, Switzerland. I'm working about viruses for a project, and I want to make a sound installation. So I have a question, and I don't find any answer on internet. Do viruses make sound? If yes, do we know this sound? Have we a record or a record? I suppose, but not... Oh, no, I suppose not, but I'm asking just in case. Maybe I'm asking an idiot question, but I don't know much about the biological virus. So thank you for your answer, and have a good day. And the only noise that I know that viruses make is... This week in virology, the kind of viruses that make you sick. That's the kind of noise viruses make.
So she sent me this on Virology blog, but I thought this was such a cool question.
It is.
Right?
It's a great question. I love it.
It is. So how would you even start to set up a recording
here's
device?
the way
You need a really small microphone.
Yeah, you need a small microphone.
A nanophone. A nanophone.
Viruses.
Exactly.
So I figure all molecules vibrate.
I know the sound of noroviruses.
Ooh.
You could have some fun with this.
All molecules vibrate, so they must be emitting sound waves, but they're very, very quiet. So I don't know how you'd record it.
I wonder if you could listen to cellular metabolism.
Viruses.
So I just responded to her by email. I said, "That's a great question, and the real question is, what key is it in?"
Yes.
Give S for sick.
I don't know. I said, we know viruses don't have color because they're too small, right? But sound, yeah, they must make some kind of sound. So you're not an idiot. It's a great question. Just make up something.
Yeah.
I mean,
make up something.
Why not? Okay.
And you could go with the sounds of various symptoms. Those would certainly be sounds of viruses.
Yeah.
But
yes,
that's probably not quite what you want.
No.
The other thing is a lot of people have done various musical interpretations where they take the genome of something and convert it
right.
to musical notes.
It's good.
Perhaps the rhinoviruses could tag on to the sound of mucus.
Yes.
Kathy.
Bill writes, as an appreciation of keeping all your shows spinning, and he sends a link to a YouTube, which I admit I didn't look at, and I'm afraid to now because it'll play over the
It's a guy with sticks, and he puts plates on each one.
Plate spinning.
Oh, cool.
And they're all spinning together.
Right.
He gets up to 8.
Yeah.
It's pretty impressive.
Okay. I always, during every TWiV, I have to start an email to myself of all the links that I have to go look at later.
Do you ever do it?
Sometimes.
Yeah, it's a lot to do. There's a lot of stuff. Rich, last one.
Bob writes, I ran across this by accident today and gives a link to a Coursera course on Zika, in the footsteps of Zika approaching the unknown. That's got, it's this MOOC, what is that, massively online
Open courseware.
Open courseware. Had been produced by the University of Geneva, Institut Pasteur, University of Paris Descartes, and Centre Virchow-Villerme. So it's got 5 different professors in it. That's all about Zika.
How about that?
Love your show. Listening to TWiV 412 right now.
Neat.
Bob.
Yes, it is the only Zika course on Coursera.
Okay.
Should I
Are there any immunology courses on?
Yeah, there's none.
Okay.
Nada.
Gee.
Should I do
hard.
Juan's email, or are we stopping there?
Stop there.
Okay.
Did you not do one?
I didn't do one in that rotation, but I'm not hurt.
I'm skipped. You mean I skipped you?
You skipped me.
It's because I don't have you on my list. Did I skip you for the last hour?
No, I think you skipped me in the last 2 cycles of email.
So sorry.
That's why I thought maybe Kathy hadn't gone, because it seemed like somebody hadn't gone,
sorry.
but I guess it was me.
All right. All right, we'll get you next time.
All right, not a problem.
Let's do some picks,
I
Alan.
say plenty on the show. It's not a big issue.
Alan, what do you have a pick for us?
I do have a pick. Let me get scrolled to it. All right. This is a little bit strange because I have not actually had a chance to evaluate this yet, because it has not actually been shipped yet. So this is a game. It's called Lab Wars. It's a card game, card and board game. And they did a Kickstarter. I didn't contribute to the Kickstarter, but apparently they raised $65,000 this year to get this game printed and manufactured.
Wow.
And they are now doing that. They've sent it off to the printer. They apparently wrote the checks, and things are happening, and they're expecting to ship it in February. I have pre-ordered it, so I'm on the list. It's £19, which with shipping.
Whiz.
Shipping is very reasonable from the the UK to here, so it worked out to about $30. And I've got this thing on the way, and it looks like it ought to be fun.
19 pounds is how much it costs, rather than
not how much it
weighed.
weighs. Yes, no, that would be quite a game.
That's the lab.
Yeah. So, and these folks seem to know what they're doing with game design. They look like the kind of people who would design a good game, and the theme of it is that you're, I guess, a PI, and you have to through building up your own lab and sabotaging the labs of others, you try and succeed academically.
Yeah, this looks good.
Yeah.
What is a blind reveal mechanic?
I'm not entirely sure.
So it says during the beginning of each round, players may play one of their 5 characters, their research team, in a blind reveal mechanic.
Right. So I think it maybe is that you put them down on the table, but face down.
Yeah.
And so you've committed to that particular play, but you haven't revealed what the play is yet.
Okay.
I think it's a reference to poker where they have blinds.
Maybe.
Right.
And you can't see them, but
right.
they're there.
That's the one card game mechanic I can think of that might be called that.
Yeah.
That looks good. You know, about a year ago, some company in Germany said, we'd like to send you this science game. It's called The Art of Science, and it's a card-based game. Mm-hmm. Would you play it and review it on TWiV? And I said, sure. So I saw it this morning. I never opened it.
Oh, dear.
In my office.
Oh, dear.
Nobody in my house wants to play it. So maybe the next time
bring it in here.
We're together.
Yeah.
Yes.
We should all play it. That would be fun to play it.
Play it online.
I've started getting back into board and card games lately.
Maybe why don't we play it and record our playing it? That would be fun.
Oh.
Yeah.
Oh, there's a whole channel of stuff that does that.
Yeah.
They have fancy gaming tables. So my brother goes on the Jonathan Colton cruises, and they have all kinds of gamers come to those, and so that's how I've seen some of those channels, video channels, playing games. Yeah,
I think it would actually be really cool if we could set up maybe at ASV.
Yeah, that's something.
Have like a game lounge, maybe if we can get a conference room one of the evenings or something, and just have a bunch of different board games and set them up, and people can come play. And this would be one that set up that way.
Well, we'll all be there.
We will be.
Dickson won't be there, of course, but the rest of us will.
Call it Game of Moans.
Rich Condit, what do you have?
Just a little item I found in the New York Times titled "You're a Bee. This is what it feels like." And this is an interactive, popular science thing that spins off of an article that they actually linked to in the Journal of Functional Ecology, where it's a review article where they're talking about the sort of coevolution of bees and plants with respect to a particular problem, which is that if you let the bees go completely crazy, they'll actually compromise the pollination of plants because they'll just rip off all the pollen. Okay. And so apparently, according to these guys, or at least according to this New York Times write-up, one of the reasons that nectar evolved was to distract the bees so they don't rip off all the pollen. But I like the way it was written, and I like the picture of the bees loaded down with pollen and stuff.
Yeah,
So
it's a cool series of things. I think I got distracted and didn't finish all the choices in the network, but it was fun to do.
It's a choose your own adventure.
Yes.
Nice.
Dickson, what do you have?
I have something called Star in a Jar fusion reactor.
Oh, yes.
It's a tokamak type of thing that they're talking about here, but apparently there's one in Germany that's actually about to actually produce more energy than it uses to start up. So, and they're in cahoots with the group at Princeton, which had one of the first tokamaks, and I'm a big fan of tokamaks. I love the concept, and I love the potential result that they would get from it. And as they mention in this write-up, it's a radiation-free source of energy because it fuses rather than splits. And as a result, when you shut the machine off for whatever reasons, if it's going out of control, nothing bad happens. It just cools down. And that's the end. But they're very, very close now to actually achieving the level of energy production that's possible by looking at what happens inside the sun. So I'm jazzed about this. I really am. It's taken a long, long time to get to this stage. And the U.S., in all their infinite wisdom of funding physics research, decided not to fund tokamak research. So the Germans, of course, took up the lead.
And by the way, that's tokamak.
Tokamak.
With
tokamak.
K's, not to be confused with Topamax, which is a
or token.
nerve pain drug.
Right. In case the toe-like receptors are being...
Should I tell everyone that your original pick was something you had picked 2 episodes ago?
Well, that was the original
I think you just did.
No, I didn't. No, I didn't.
He sent me
No,
a
but
pic.
Vincent just did.
Vincent pointed out.
I pointed it out.
And
yes.
rightly so, because I do recall having picked it, but I don't recall it was that
I
recent.
think it was funny.
I don't.
It was genuinely funny.
I'm concerned.
Because he sent it and he said, did I pick this before?
I'm very concerned.
Kathy, what do you have?
I picked something that came out of email that we got this week from another faculty member in another department who had been taken in by a so-called predatory journal. Call me naive. I wrote an invited review for such and such journal. Everything looked fine, page proofs, etc., until I got invoiced. I wasn't informed of any page charges, and I won't pay. And then I checked if those papers in this journal are indexed by PubMed or the Web of Science. They aren't. So then someone else wrote back and said, there's a list of possible, potential, and probable predatory journals. So I thought it was worth including that for anybody who gets some kind of review request if they want to see if the request is coming from something that might be predatory, they can just click there.
Yes.
It is a very long list.
It's an extremely long list. I just glanced down through the first few, and some of the titles are actually pretty hilarious, like Acme Intellects. And the Advanced Science Journal,
right?
I thought was pretty fun, and Actual Problems in Economics.
Really?
Unbelievable.
Yeah.
Anyway, that's just in the A's, but yeah, it's a long, long list, and hopefully it will be well indexed by Google, and maybe people won't get taken in by this kind of thing.
I'll quote Dickson Despommier: "Successful systems attract parasites."
That's exactly exactly right.
All of them.
Exactly.
My pick is an article in Stat by Sharon Begley. It's called "CRISPR Court Hearing Puts University of California on the Defensive." So the hearing over the CRISPR patent has begun.
Oh, my.
And the first paragraph: if the number and tenor of questions addressed to each side during oral arguments in the CRISPR patent fight is any indication of what patent judges are thinking, the University of California has a steep hill to climb if it wants to overturn the 2014 decision awarding key CRISPR patents to the Broad Institute of MIT and Harvard.
Oh, my.
So they were awarded the patent, and Berkeley doesn't like it because that is where Jennifer Doudna is, who has done very important groundbreaking work. So there's a lawsuit. And there it is. You can read about it. And that's the last I'll mention that.
What a waste of overhead money.
Indeed.
Yes.
Indeed. Now, we had 2 picks already, I believe. We had Katreya and someone else that Rich had read. And then let me read you 2 more.
Up at the top, wasn't there in the follow-up?
Yeah, there was when I moved it down to here. I didn't read it yet. I have 2 here. One is from Pritesh, who writes, "Hi, TWiV team. Here's my listener's pick." It's an article. The original article is in Current Biology, and he sends a link to a Science magazine article. "Virus found in child mummies suggests recent rise of deadly smallpox." We miss Dr. Condit at the University of Florida. Hope he joins us sometime for Viroholics and Journal Clubs. Pritesh is a PhD student there.
I miss you guys too, Pritesh.
Rich, do you know this?
Good place to be.
So they recovered a whole genome sequence from a more recent a mummy,
yeah, it's
I think, in Lithuania.
Like an 1700s.
Yeah.
Something like that. I was not aware of this, and I looked at this, and this is actually quite interesting because they were looking for something else in this mummy. They were looking apparently for a polyomavirus or something like that. But these, in their sequencing, these variola sequences, that is smallpox virus sequences, came crashing out.
Wow.
And they had enough sequence information so that they were able to do an entire assembly. And when they did the phylogenetic analysis on it, yes, it's considerably. I mean, all the other sequences that we have are pretty recent viruses, right?
Yeah.
And this one it looks older, yes, but based on that and other phylogenetics, it suggests that variola, as we know it, came on the scene much later than was previously thought, maybe in the 15th or 16th century, rather than as has been thought back as far as the Egyptians, like 3000 BC or something like that. But those descriptions, I mean, that notion that it dates back to the Egyptians is based primarily on historical writings describing a disease that we interpret to be smallpox. This is real data. Now, whether the phylogenetics is telling us the real story or not, I don't know. But it certainly is it's interesting to think about. And apparently this is not the first time that this notion that smallpox is an ancient disease has been questioned.
Yeah.
It's been challenged before, I think, based on the existing phylogenetics and on the notion of what they say here is you can't really, in a clinical description from hundreds of years ago, you can't necessarily discriminate between smallpox and variola and chickenpox. But and then I started thinking about it, and I thought about one of the historical records that I think is probably screaming with smallpox is the conquest of Mexico.
Right.
I have a hard time believing that that's not smallpox that Cortez brought to the Aztecs. But that's still, I looked at, that's 1520. So that's not inconsistent with this. And I don't know what sort of, I'm not enough of a historian. I need to go read, what is it? Guns, Germs, and Steel.
Yep.
Yeah.
Right.
And put together more of this. But this is very interesting. This is very interesting.
Yeah, it's cool. There's a lot of virus in this mummy, and they got lots of good sequence, apparently.
So my mummy done told me.
What's that?
My mummy done told me.
Yes.
I also want to point out that part of this is comparing the sequences of 46 different strains of variola that had been isolated, which obviously this has been done over time, but that would not have been possible had we not kept those strains around.
We destroyed the stocks. Right.
So from this mummy, it would just be nucleic acid fragments? It's not the virus itself, or
correct? They talk about virus, but all they're doing is nucleic acid. And I didn't look closely enough at the article or anything else to know whether anybody actually had a look for virus itself. But they're just dealing with nucleic acid.
How long does smallpox virus survive on fomites?
Long time. It's tough stuff.
I thought. So do you think that's how it came over to the New World? Because I can't imagine Cortez's men actually being sick from smallpox.
Oh, they might have been.
You think?
Yeah, could have been. I don't know. My guess is there are records of that.
Yeah, there's a science article that Pritesh linked us to, and they say the sample was rich with variola, the virus that causes smallpox, which isn't really correct.
Yeah, that's a misstatement.
DNA.
Right.
Rich with DNA.
Let's go. Let's know the difference between virus and viral DNA. All right, our other pick is from Drake, who says, since many who listen to this netcast are interested in low-cost lab equipment or only get their science fix in the kitchen/garage, I figured I'd share something that is on my holiday wish list. The Spinzall is a low-cost centrifuge designed for restaurants, bars, and advanced home cooks. It is interesting from a technical point of view since it is portable, self-balancing, and has a pump for continuous feeding. My main interest in the centrifuge would be for making herb oils, pea butter, fresh olive oil, and advanced soft drinks or cocktails. Thanks for all the time you spend on this netcast. And he provides a link to this Spinzall.
Which I gather is kind of a glorified milk separator.
Oh, this is a crowdsourcing type of thing.
Right. It's a crowdsourcing campaign to build this thing. I'm not sure how many Gs you can generate with a milk separator, which those have been around for decades, and you can find them on eBay and old farm stores. But it's the same principle, I think, where you feed the liquid in continuously, and then you have a spinning chamber. Milk separators, the ones that you might have in your kitchen, are classically hand cranked, but of course they can be motorized up to industrial scale at dairies.
They've raised over $200,000.
Yeah.
But they need to reach their goal.
600,000, yeah.
Yeah. I don't know if they'll make it.
Oh, they'll get the TWiV bump.
Right.
Hey, what is pea butter?
Right.
Butter made with peas?
Yeah, some kind of
I guess so.
pea sauce, yeah.
That's PEA, by the way.
Right.
Yeah.
We're all going to be conscious of this for a few episodes.
Yes.
Dickson is Dickson.
It is.
MOUSE.
This episode of TWiV 419, you can find it at iTunes, microbe.tv/TWiV. If you have a smartphone, not a Galaxy Note 7, but other kinds, you can grab podcasts for free on any number of podcatcher apps. They're all free. You just subscribe so you get them automatically. Please send your questions and comments to twiv@microbe.tv and consider supporting our efforts so that we can travel around and unify at various meetings and do all sorts of cool stuff for you. You can go to microbe.tv/contribute. We have several ways that you can help us out. Dickson Despommier is right here, but he's also, right here meaning Columbia University in New York City, but he's also at TheLivingRiver.org. He's also at ParasitesWithoutBorders.com.
Right.
Thank you, Dickson.
Great time.
Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Rich Condit is an emeritus professor from the University of Florida in Gainesville, currently residing in Austin, Texas. Thank you, Rich.
Excellent. Very well done. Always a good time. Thank you.
Alan Dove is at alandove.com. You can also find him on Twitter. Thank you, Alan.
Thank you. It's always a pleasure.
And I am Vincent Racaniello. You can find me at virology.ws. I'd like to thank the sponsor of this episode, CuriosityStream. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.